Fantastic. It's 10 o'clock. Welcome to PAR Foundation and the webinar series that we're initiating this fall. Today, I'm very pleased to announce that we're starting our webinar series. We have built a program over 9 sessions from today until December 6th, and you will be able to listen in to 25 plus interesting speakers who will talk about insights and their own research to prevent antibiotic resistance. Our main focus is really to put light on that preventive actions to stop the spread, and we do that together with all these experts during the fall. To start with, I just want to show you today's agenda. We have 3 really strong speakers on the agenda today. My name is Cecilia Dahlström. I'm the Secretary General at PAR Foundation, and I will be moderating the session today. With me, I also have Gabriel Oke. He's working as our communication manager, and he will be tracking and following the seminar on the back end. He will take care of questions. He will also be your main contact during the fall when it comes to our webinar series. To start with, I just want to give you an idea of PAR Foundation and who we are and why this is important to us. Our aim is really to help eradicate the spread of antibiotic resistance. And we do that by engaging and supporting researchers and passionate teams that work with educational initiatives in the area of prevention of antibiotic resistance. We've done so since 2017, and since the start, we have funded 25 projects that have been working across Europe, but also in Africa, Asia, and this year we're funding the first Latin American project. We have a community of 1,000 plus engaged, passionate researchers within the field that we talk to on a regular basis, and this group will join us during the fall, but are also part of grantees that has received the 16 millions that PAR has distributed so far. We like to think that we make a difference. 2022, we focused on educational initiatives and wanted to spread awareness around AMR. This year, we want to move into action. We want to take preventive actions to stop the spread and making a difference to work towards global health. Right now, we have 8 active programs running, and we're about to initiate 8 new projects in the coming 2 weeks. So that is a short introduction of PAR and who we are. Without further ado, I'd like to hand over to our first speaker today, Zeenatuddeen Mohammed. I will leave it to you to introduce yourself and then tell us about your project today. So, Zeenatuddeen, please take it away.
Hello, good morning.
Good morning. Nice to hear you.
Yeah, I am Zeenatuddeen Mohammed, a graduate of microbiology and a holder of an MSc in veterinary public health and preventive medicine. And I think I would like to say that one of the reasons why I went to do my MSc in veterinary public health and preventive medicine was because I was actually interested in how infectious diseases were transmitted from animals to humans. That's just, in short, zoonosis. And I think we cannot do that without having little knowledge about some of these areas and how we can actually collaborate. And currently, I think working with the Environmental Health Council of Nigeria, I have also gained an insight on how the environmental health, human health, and veterinary health can come together to fight some of our current public health challenges, including antimicrobial resistance. So thank you. My presentation will focus on environmental dimension of antimicrobial resistance.
Fantastic.
To start with, let me go over the agenda for today, the ones I will be focusing on. I will basically talk about introduction, resistance evolution in the environment, pollution as a driver of antimicrobial resistance, environmental transmission of antimicrobial resistance, and how the environment is reflecting a clinic, so to say, and then environmental actions needed to reduce the burden of antimicrobial resistance. And then I will round up with a short conclusion. Next. So antimicrobials are agents intended to kill or inhibit the growth of microorganisms. So when we talk of antimicrobials, we don't only mean drugs or antibiotics that are prescribed in the hospital. The agents can be chemicals that can inhibit the growth of the microorganisms we don't even target. Antimicrobial resistance occurs when microorganisms such as bacteria, fungi, parasites become resistant to antimicrobial treatment to which they are previously susceptible to. Resistance can be an evolutionary response by microorganisms which genetically change their makeup in such a way that they are no longer inhibited or killed by antimicrobials. Microorganisms have different ways of achieving antimicrobial resistance. Some of them change their target site by altering it so that the antibiotic that is prescribed or used to treat them can no longer work. Some of them have efflux pumps through which they can pump out the antibiotics that have been injected into the system, and various other mechanisms that the microorganisms use in order to become resistant to the antibiotics that are used for the treatment. Next slide. Next slide, please. Okay, so resistance evolution in the environment. Normally, microorganisms, let me focus on bacteria. They are ubiquitous in the environment, which means they can be found everywhere in the soil, water, and air. So they can develop resistance naturally in the environment when they interact between themselves and other microorganisms. So there can be horizontal or vertical transmission of genes that can confer resistance to this microorganism, even in the absence of an antimicrobial compound. These are naturally occurring processes in the environment. So resistance can also develop due to selective pressures from antibiotics, fungicides, parasiticides, even heavy metals and other compounds that are exposed, that the microorganisms become exposed to. So this selection processes will now lead to antimicrobial resistance developing in these microorganisms. Antimicrobial resistance can also arise from mutations in the genes or genome of the bacterium, and from the uptake of foreign DNA from other bacteria that are coexisting with them in the environment. So resistance is actually comes in various ways, and there is no particular demarcation that we can understand, okay, this microorganism became resistant because of this factor, only factor. So normally when an antibiotic is prescribed, if the bacteria is a non-resistant pathogen, the antibiotic will be able to clear the bacteria and the person becomes healthy within the expected range of time. But for resistant bacteria, you treat, and then the infection is still there, or it will become mild and later comes back to an acute stage. So even in the human system, there can be resistance when exposed to these antibiotics or when not used as prescribed or as it should be used. Next slide, please. Pollution as a driver of antimicrobial resistance. Although the natural production of antibiotic molecules most likely contributed to the evolution of antimicrobial genes, it is not responsible for the rapid evolution and expansion of spread of this resistance. Antibiotics reach the environment through various processes, such as urine and feces from humans and domestic animals, improper disposal and handling of unused drugs, effluents from pharmaceutical industries and other manufacturing industries, and even from agriculture, the use of manure and other chemicals in the environment during farming or treatment of animals. All this can make antibiotics to be present in the environment. The concentration of these antibiotics and the abundance of these antimicrobial genes often correlate in environmental samples. There are various researches that have shown that the concentration of antibiotics in the environment correlates, very, very strong correlation with the antimicrobial genes that we are having in the environment. Therefore, environmental contamination with fecal bacteria provides physical contact and therefore increase the opportunity of gene exchange between resident environmental bacteria and the bacteria that are adapted to the intestinal tract of humans or domestic animals. This makes the environment a very, very important factor when we are talking about preventing antimicrobial resistance. I see the environment as a mixing point where microorganisms from humans, from animals, and those that are originally resident in the environment, where they mix and can exchange their genes. So therefore, some genes that were not present in some human pathogens can now be acquired during mutation and other processes. And therefore, in the environment, these antibiotics, heavy metals, biocides, and other drivers of antimicrobial resistance will now accelerate the development of antimicrobial genes in these microorganisms. Fortunately, for us in the low and medium income countries, such as Nigeria, the experience is that most of the states don't even have waste treatment plants, and even handling of waste is very, very poor. So sometimes the microorganisms were resistant before they even infect us. And that is why in the hospital, they will be treating, and then most of the time the treatment will prove abortive. And even if the treatment is successful, I think it will still circulate in the environment because it's not actually something that can be gotten rid of. It's not antibiotic related issue. So hand hygiene and other things are what is critical. Next slide, please. Environmental transmission of antimicrobial resistance. The environment can provide a route of transmission of resistant bacteria to colonize or infect humans. There is always an exchange of antimicrobial genes and antimicrobial resistant bacteria between food animals to humans, from the environment to humans because of interaction in the natural ecosystem. Therefore, transmission events become very, very common and is probably the case in many low and medium income countries. With inferior infrastructure for handling fecal waste, environmental transmission may have profound effects on the overall resistance situation. Therefore, correlative analysis have suggested that overall sanitation and waste infrastructure is a better predictor of national burden of antimicrobial resistance than is reported in the case of use of antibiotics. It has also been shown that fresh produce often carry various resistant bacteria with diverse mobile genetic elements. Therefore, the safe use of human and fecal and animal fecal matter, such as manure, sludge, and other effluents from wastewater treatment plants on farmland, is warranted both to control the transmission and also to control resistance evolution in the environment. Next slide, please. The environment reflecting the clinic. Although the environment contributes to the problem of antimicrobial resistance, both during the evolution and also as a transmission route, it can also provide a means of managing antimicrobial resistance. Therefore, several studies have highlighted the possibility of using sewage monitoring, for example, in conjunction with clinical surveillance of resistance, either using phenotypic analysis of isolates or analysis of the antimicrobial resistance genes using PCR and other methods. So resistance surveillance in sewage can provide several benefits over traditional surveillance using antimicrobial use in the hospitals, for example, and more research and extensive benchmarking of the potential and limitations to bring this surveillance strategy into practical use. I think if you can incorporate environmental monitoring into the surveillance of antimicrobial resistance in the hospital, we can have a better way of managing antimicrobial resistance in the environment. Next slide. So the environmental actions needed to reduce the burden of antimicrobial resistance. First, I think is public education and awareness, just like Power Foundation has been doing. And I think in 2022, my best time was when I discovered Power Foundation. And I think I have learned a lot. I was always going through their page because that was an issue that I was battling even in my place of work. Anytime I bring the issue of antimicrobial resistance being in the environmental sector, they will think this is for doctors, this is for the human health sector. So I think public education. And then when we are doing public education, one issue I have is we cannot improve awareness and understanding without effective communication and education and training. Because when the communication is not effective, I don't think people will understand what we are saying. Just like in my place of work, most of them are professionals, but to understand that the environment has a role to play in antimicrobial resistance has been a battle I have been fighting for so long, and I think I'm making progress. So public education on AMR is very, very essential. And then we need to enhance environmental governance, planning, and regulatory frameworks. To do this, I think the ministries of environment, especially in developing countries, need to implement all national action plans. I'm happy that Nigeria has, we have our action plan. The implementation is the problem, but we are still going forward. And then the implementation cannot work in isolation. We have to work, we have to make it a multi-sectoral approach, because anything you do in isolation, I don't think we will achieve what we want to achieve. And then we need to implement at the grassroots, because mostly in the cities, at least we maintain a very good hygiene. And then in the low-income countries, the cities that suffer are mostly the villages. We also need to identify and target priority AMR relevant pollutants. For example, human waste that is mostly disposed. Sometimes you will see a dump site, and by the side they will be disposing human waste or animal waste. I think those actions need to be stopped, and then we need to have good regulatory frameworks that will help checkmate these activities. We also need to improve reporting and surveillance and monitoring systems, because currently, I don't know, maybe in some countries, but in Nigeria currently, we don't have any reporting system, and we don't have a working monitoring system. Surveillance is ongoing, but mostly it has been restricted to the hospitals, which I think is not enough. We also need to prioritize financing, innovation, and capacity development. The government cannot do it alone. Thank you, PAR Foundation, for supporting some of us that are interested in fighting antimicrobial resistance. And then innovation is key. We cannot just do it the way it has been done everywhere. Sometimes our environments are unique, so we have to get strategies to do that. So in conclusion, the environment has a role to play in both the evolution and transmission of antimicrobial resistance. Possibly more so than has generally been recognized. A remaining and pressing knowledge gap is our limited understanding of where and under what circumstances the critical steps occur that lead to the emergence of new forms of resistance in clinically important bacteria. Thank you very, very much for giving me the opportunity to speak. You remain Zeenatuddeen Mohammed. Thank you once more.
Well, thank you, Zeenatuddeen, and thanks for giving us the perspective from Nigeria. I think that is very interesting. And I noted that you had a very interested companion as well,
Yes.
who's hopefully becoming an ambassador for the work that is needed within the field.
Hopefully.
Thank you so much.
Welcome.
I will then move on to Ibrahim. Ibrahim, would you like to introduce yourself? And then I will stop share my screen and you can share your own screen. But just 2 words on yourself.
Okay. Thank you very much, Cecilia. I am Ibrahim Yusuf. I'm a microbiologist by training, a medical lab scientist. I work with the Department of Microbiology, Bayero University, Kano. I have been working and teaching, conducting research and training in the field of antimicrobial resistance. I have trained quite a number of students at both graduate and postgraduate levels. And I have been engaged in antimicrobial resistance related projects in Nigeria, as well as outside in Nigeria. I have served as several consultant to develop antimicrobial resistance surveillance system in the environment for the Nigerian and the other partners.
Thank you.
Thank you.
So I stop share and then I ask you to do sharing from your end instead. Thank you.
Okay. Okay. I'm sharing from the okay, so sorry. So can you see it now? Can you see my screen?
Yes. Absolutely.
Okay, so good morning, everyone. I'm presenting on the hotspots of antimicrobial resistance in Nigerian environmental media.
And I will interrupt you. You're showing the last slide right now, so you have to go to the first slide.
I'm in the first slide. I don't know.
If you click on the first slide, I think you will be able to see it. Scroll.
Okay, is it there?
You're showing the last slide, so you have to click on your first slide.
Double click on the first slide. Double click on the first slide. Under the slide column.
Okay, so I think let me do it this way. Yeah. Sorry. Okay. So I'm sharing again.
Let's make a new try.
Yeah, sorry. Yeah.
There you go.
Yeah, now it's okay?
Now you're on the first slide.
Yes, yes.
Okay, okay. So sorry for the interruption. So my presentation will be on the hotspots of AMR in Nigerian environmental media. I believe the first presenter has been experienced in the field with the environmental health. She has summarized the situation in Nigeria. I think my presentation will build on what she has presented. So my portion of the presentation will be on the hotspot of AMR in Nigerian environmental media. So we give some few background to this, how it gets started. Nigeria, like any other country, has one health strategic plan in place. A strategic plan that tries to integrate human, animal, and environmental health in fighting antimicrobial resistance.
I'm going to interrupt you.
Okay.
You have to go to the next slide.
Yeah, I'm there. I don't know what happened. I'm on the second slide already.
Mm-hmm.
So it's not changing?
It is not changing, no.
You have to double click. Have you double clicked on the slide you're on? I can see that it's highlighted in yellow.
Yellow, green. It's green. It's highlighted in green already.
Oh, something is wrong then. I think you need to maybe share again. I don't know.
So what do you suggest? I stop sharing and share again?
Because
or can Gabriel share it from his side? I think you have it.
Oh, I think that's better. That's better. I'll do that.
Yeah. So
just a minute.
Okay, so I'm stopping.
Mm-hmm.
Hello, can you see my screen now?
Yeah.
Absolutely.
Yeah.
The color, good. So is this the slide you're on?
Yes.
Or the next one? Okay, good.
Yeah.
Good. Go ahead.
Okay, so can you make it a little more presentation mode then?
Can you see it?
Yeah, I'm seeing it.
Presentation mode. You're not in presentation mode either.
I'm not in presentation mode?
Yeah.
Wow. I think the same issue that you are. Because I'm on my screen now, it's presentation mode. What of now?
Yes.
Now it is.
Okay,
yeah.
Good. Thank you.
Okay, so thank you. So I think Gabriel will control the sliding. So as I was saying before, Nigeria have one health strategic plan in place, which integrates human, animal, and environmental health together in fighting antimicrobial resistance. Significant efforts are in place in human and animal sectors. As far as surveillance system is concerned, they have an active surveillance system for antimicrobial resistance. But then the environment has nothing, as Zeenatuddeen has mentioned earlier before, that AMR surveillance is not available in the environment, probably because environment has more complex situation whereby you are not dealing with one aspect of AMR, unlike human and animal, where you only deal with the resistant bacteria alone in most cases. But in the environment, we are dealing with multiple issues. We are dealing with the antimicrobial resistant bacteria themselves. We are also dealing with antimicrobial residues, the residual antimicrobial agents in the environment, as well as also antimicrobial resistant genes in the environment. In the case where some organism could not be cultured, so you can know of their fate by detecting the antimicrobial resistance genes in the environment. So next slide, please. So we are on this situation until sometime. Next slide, please. Okay, thank you. So sometime in 2019, the Fleming Fund came, and to be able to address the issue I mentioned for lack of surveillance system in environments, one of its key objectives was to lay the foundation for the establishment of AMR surveillance in the environment. Then we can start the work then. And one of the questions we need to answer then, because we are starting from the scratch, is what are the entry point of AMR into the Nigerian environment? Then we couldn't get the answer, but we were able to get through some survey and some interview, we were able to identify some key entry point into the AMR. Then we don't know the hotspots of AMR in the environment then. So we need to answer this question too. Then we searched, we couldn't get any data on ground. Then we have to leverage on other data from all over the world, because the key hotspots may be general to all nations, but we have some specific ones. So next slide, please. Okay, so before I move on very quickly, the hotspot, as far as this presentation is concerned, means regions which are known for high species or high number of antimicrobial resistance. Will it be antimicrobial resistant bacteria? It will be antimicrobial resistant residues that have higher concentration of residual antimicrobials in the place, or a location that have higher amount of antimicrobial resistant genes, which is what we are trying to look for as far as this as far as hotspot is concerned. So the selective pressure antibiotic have on these organisms is what? When we have high concentration of antimicrobial agent in a place, this selectively allow the proliferation of resistant strains and clear away the susceptible one. That is what we mean. So you can see the hotspot in the center was surrounded by a lot of things. The antimicrobial concentration present in the place, the antimicrobial resistant genes on another side, as well as the pathogenic microorganisms themselves. So these are what we are dealing with. So now, in the next slide, we're now able to understand and get information from the key hotspots, which we get from the literature. The key hotspot for AMR in the environment include wastewater treatment plants. This is general, but as mentioned from the previous speaker, she said majority of these wastewater treatment plants are not available in low and middle income countries, including Nigeria, in many places. So that is one issue. So wastewater system, that is the sewage system in the environment, also serve as a good hotspot where you can get antimicrobial resistant microorganisms there. But are these always available and working functionally in low and middle income countries? This is the question we could not answer as far as then. The next option, the next hotspots was now pharmaceutical manufacturing sites, the location where factories that are producing antimicrobials are located are also a good hotspot where you can get highly resistant microorganisms. as well as food and animal production sites because of the number and level of antimicrobials being used in animal production, especially in our setting here. So you do have
Ibrahim, if you can hear me, we can't hear you. Mr. Ibrahim Yusuf, if you can hear me, we can't hear you. You can off your video so that the okay. You can off your video so that the internet will be strong.
Okay. So can you hear me now?
You can hear you now. Go ahead.
Hello, can you hear me? Oh.
We can hear you, but I will advise that you off your video so that the internet will be enough to cater for the audio only.
Okay. So is that okay now?
Yes, go ahead.
Okay. So quick, next slide, please. Okay, now I'm moving into the AMR hotspot in the environment, which is specific to Nigeria now. The previous slide is general all over the world. That is what is available in the literature. But now I'm moving into what is specific to Nigeria. So since the beginning of the project, one of the recommendations is to kickstart the process of providing data, in-house data on the antimicrobial resistance in the environment in Nigeria. So the project, a lot of projects have started since 2019. Up to date, some are still going on. So we are able to identify key 6 for now from this region. Other regions have different hotspots. So we are able to gather these 6 regions based on the data provided and the data gathered over time. One is the municipal drainage system as one of the key hotspots of AMR in Nigeria. The second one is the municipal solid waste disposal or destruction sites. That's another key important area where we get large amount of antimicrobial resistant microorganisms, ARGs, as well as the residues. The last, number 3 is the industries waste and effluent disposal sites. Then where drug disposal or destruction site, expired counterfeit drugs or confiscated drugs were being destroyed. The sites and the surrounding are now becoming a very good hotspot for the development of antimicrobial resistant microorganisms. Number 5, soil along industrial effluent sites, as well as shared surfaces in the very crowded area. So I'll take them one by one pictorially so we can see what I mean by that. So next slide, please. Okay, so number 1 is the municipal drainage system. In ideal situation, the waste that leaves the residential area need to be connected, channeled to the waste disposal site where it will be treated and used again. But notwithstanding, in some of the low and middle income countries, the situation is not like that. We're able to sample quite large number of sites where the pictures as you are seeing are some of these situations. These are residential areas, and the waste from the residential area has no any proper channels or drainage system where it can move on. So they get stagnated there and stay there for years. So it only gets cleared sometime once in a month or thereabout, depending on the situation on how it disturbs the populace. So samples around this area show that we get different and high number of antimicrobial resistant microorganisms, a quite large number of antimicrobial resistant genes in those areas. Seasonally, I mean persistently, always when you check there, you always get them in high number including some of the key indicator organisms, which are the extended spectrum producing antimicrobial bacteria such as E. coli. Yeah. So this is number one. So next slide, please. Yeah, so you can see another situation. You can see the houses behind around the surrounding area. So the waste that come from those houses and the neighborhood comes into this stagnated place, and it has been like that for years. So the waste containing antimicrobial agents from the domestic area, from all other places, stagnate here. So the microorganisms surrounding this environment, either in the water or the surrounding soil or surrounding plant, if there is any, do have high resistance to great number of antimicrobial agents, especially those that are used in the clinics, in the hospital, as first-line antibiotics. So next slide, please. The second one now is the waste disposal site, which you'll see in the next slide. Municipal waste disposal site. In ideal situation, the waste generated from the houses is supposed to be a kind of being a system that gets either recycled or disposed in a proper manner. But in this situation, in most of the low and middle income countries, we don't have such a good system in place, especially unless for some bigger cities. So situation like this, where the waste are dumped alongside just nearby a residential area, is a very good hotspot for antimicrobial resistant microorganisms, antimicrobial resistant genes, as well as high concentration of antimicrobial agent, including heavy metals, biocide, and all this. So sometimes they do combine with this the water system and the waste combine together. So we're able to sample quite a number of these over years, and we're able to isolate from individual component of the waste as well as from the soil, and we're able to determine what are the contributors of AMR, ARG to the disposal site, and what are the factors that fuel their transmission from this location to other locations elsewhere. Next slide, please. So you can see from here the component of the waste are hereby highlighted. You can see wastewater, diapers all come together, waste food, carcasses of animals all get dumped in these sites under their nearby residential areas. And quite a number of human activities that can fuel their transmission are also available. You can see animal scavenging, you can see winds, you can see open burning and runoff, and as well as human scavenging. All these are available, and they quickly transmit this microorganism from one side to another. So let me move quickly to the next slide, please. Number three. So we've put some of these things together in the paper, although it's still under review, but very quickly, we hope very soon it will be available for you to see what is available in the Nigeria's waste products, waste disposal site. Next slide, please. So okay, so industrial heavy metal disposal sites. These are some industries dispose their heavy metals waste directly into the environment without any prior treatment. So microorganisms from different locations around this site are also highly resistant and becoming a very good hotspot for the spread of antimicrobial resistant microorganisms. Next slide, please. Number 5 is drug destruction sites. We sample because of drug exposed to fire. Some of them become only destroy the container and expose the active ingredient into the environment. Runoff takes it from the initial burn site to up to 50 meters away. We're able to sample along the meters, and we're able to identify quite a large number of antimicrobial resistant microorganisms, as well as high concentration of different antimicrobial residue of those drugs. So second to the last, next slide. So shared surfaces. So in the hostel, in the university, or in the schools, or in the public gathering, you have high population of people using one, sharing one accommodation or small toilet or this together. So we sample as for consecutively 4 years from different sites and persistently been isolating key antimicrobial resistant microorganisms, which we link to the hygienic condition, as well as the large number of people using a smaller smaller area. So these are the key hotspots we are able to identify for now, and research is still ongoing. I think we need to put in place some strategic plan in place together with the where Zeenatuddeen works to check all this and put strategic intervention protocols in place to checkmate the evolution and the spread of this AMR from the hotspots. I think that is what I have for you. Next slide, please. I think. Yeah, this is one of the people, and thank you for your attention.
Okay.
Federico, did you want to ask a question to Ibrahim?
Yes, thank you. Can you hear me well? It's a common question that is also related to Mohammed's first presentation. So basically, we heard in these two very interesting talks that the environment is reflecting the clinics. So the fact that, for instance, there are maybe antibiotics used in animal farms, and do these and also the waste in Nigeria that we saw from Ibrahim's presentation. So does this reflect the cases of antibiotic resistance that are also observed in the clinics? That was my question.
Mm-hmm. Yeah, do you have a reply to that, Zeenatuddeen?
Okay. Even though, okay, can you hear me?
Go ahead.
Okay, yeah. Even though the question, my connection was breaking, what I had, if I had well, your question involved the use of antimicrobial in the animals, and or what we get in the environment. Do they correlate what is found in clinic? Is that your question?
Yes, exactly.
Yeah. Yes. So part of the work we are doing is to track, especially the Acinetobacter baumannii from those areas. We're able to conduct whole genome sequencing, and we're able to track some of these microorganisms from the environment far away, link them to form a cluster with those from the clinic in the hospital. And that is where the great problem was. Quite a number of them, yes, only developing resistance and acquiring virulence genes that can enable them to successfully stay and establish in the hospital. But we were able to find quite a number of them, although percentage is still low, not less than 10, and those found in the environment linked to the clinical situation in the hospital. We are several meters apart. So the correlation is there. Part of it is the waste treatment plant we have in one hospital only treat it hospital waste partially, not complete, and end up discharging the waste outside the hospital, and the waste are now being used by farmers to fertilize their crops just by the other side of the hospital. So these are available, and we are not surprising that some of these key organisms found in the environment were clinically relevant. I think in the slide I show on the paper we presented here is can say a detection of clinically relevant microorganisms from the environment, meaning those that are involved in a clinical situation are also found in the environment. But if that answers your question.
Yeah, thank you very much. Thank you.
Thank you. I think that is a good bridge over to you then, Federico. Federico is our last speaker today. I will hand over to you to introduce yourself, and then I'll stop sharing my screen so you can share.
Thank you, Cecilia, for the nice introduction. So, and it's a pleasure for me to be a speaker in this webinar. So, I'm an associate professor at the Department of Neuroscience at Karolinska Institute in Stockholm, and I'm the leader of the Laboratory of Neuroinfections and Neuroinflammation. So, I come from Italy, so I studied biomedical sciences, bachelor and master at the University of Pavia. And then I moved to the Netherlands in Groningen, where I did my PhD in medical microbiology, studying the molecular mechanism of bacterial interaction with the blood-brain barrier in meningitis pathogenesis. And then I got my PhD in 2013, and then I started my postdoc in infection biology at the Department of Microbiology, Tumor and Cell Biology at Karolinska Institute. Continuing studying bacterial interaction with the blood-brain barrier. And then I started my own independent laboratory at the beginning of 2021, and moving to the Department of Neuroscience, where we study now the molecular mechanism regulating bacterial interaction with brain cells.
And that is what you're going to talk about today. So I'm handing over to you.
Thank you.
I'll stop sharing. Please take it away.
Yes. So. Can you see my screen now? I put it in presentation mode.
It is absolutely, we do. There you go. Perfect.
So, yes. So the aims of our research is to understand the bacterial interaction with brain cells, and then use the knowledge that we generate to establish new therapeutic approaches by modulating and blocking these interactions. And in this talk, I will explain you how we are trying to establish new therapeutic approaches to fight antibiotic resistance for bacterial infections of the brain. So bacterial infections, so Streptococcus pneumoniae, also known as the pneumococcus, is the main etiological agent of bacterial infections of the brain, and the bacteria invade the brain by reaching in the bloodstream, the blood vessels that compose the blood-brain barrier, and then they interact with the endothelial cells that line the internal wall of these vessels to enter the brain. So a bacterial infection of the brain can cause meningitis, which is an inflammation of the meninges that are the layers that cover and protect the brain and the spinal cord. According to World Health Organization, Streptococcus pneumoniae is one of the main causes of death worldwide, and in particular, pneumococcal brain infections are a major burden in global health, especially for all the neurological disabilities that cause. So what are the major unsolved problems today concerning brain bacterial infections? Well, first of all, the beta-lactam antibiotics like penicillin that are commonly used to treat streptococcal infections, they have a very low penetration capability across the blood-brain barrier. And the major problem is that when they do penetrate, there is a high rate of antibiotic resistance spread all around the world. And last but not least is that currently today, beyond antibiotics, there are no available treatments that specifically target and protect neurons from the infections. So when the bacteria are in the brain, they meet neurons, which are the main cellular component of the brain, and they are responsible because they for controlling all the functions of the brain. And the bacterial interaction with neurons leads to brain damages, which are experienced by up to 70% of the survivors, which include a variety of disorders like cognitive delay, motor disabilities, hearing loss, and various types of psychiatric disorders. And all these are classified as neurological sequelae. And the major aspect of these is that they are permanent. So in the last couple of years, with my group, we discovered the molecular mechanism that is regulating the interaction between pneumococci and neurons In particularly, we discovered that neurons, because of their elasticity and motility, they expose on the outside parts of beta-actin filaments that are part of the cytoskeleton. And these are some confocal microscopy imaging showing orthogonal views in which you can see in red the lipid layer of the plasma membrane, and in green beta-actin filaments that are exposing on the outside, both on the XZ and the YZ axis. And what we discovered is that this is an image of a human neuron, and in red is the beta-actin exposed on the outside, that the bacteria here are able to bind to this exposed beta-actin. So what we did was to culture human neurons and then had an anti-beta-actin antibody on these neurons to block the binding sites for the bacteria. And we could interestingly see that the bacterial adhesion is significantly impaired compared to untreated neuron. This is the adhesion ratio of the bacteria. And then when we used a mouse IgG isotope control, we didn't see any significant difference. And then we also checked for the neuronal viability, so the index of neuronal cell death. And then we could see that there was a high cell death when neurons are infected without the treatment. And then when we blocked beta-actin exposed, then we could not only prevent bacterial adhesion, but this impaired bacterial adhesion was also reflected into the very low neuronal cell death, almost compared to the uninfected neurons. Therefore, when pneumococcal neuronal beta-actin binding is blocked, indeed neurons are protected from pneumococcal infection. Now we want to take a step forward and we want to optimize this blocking using extracellular vesicles. So EVs. EVs are lipid bilayer delimited particles that are naturally released from almost all types of cells. In our lab, we use X cells because of their reliable growth and high propensity of being transfected. So EVs can be coated with the antibodies to target specific cells, and because of their nano size, they can pass well human cell barriers like the blood-brain barrier. So for this project, we are adopting a double approach. So one is to use active modulating drugs that are these ones that are very well described in the literature. They can basically reduce the attachment points of beta-actin filaments within the plasma membrane without altering the functionality of the cytoskeleton of the cells. And in this way, they can reduce the exposure of beta-actin filaments on the outside. So we did adhesion assays, and then we could see that the single drugs, a combination of two drugs, and then in particular, a triple drug cocktail could very significantly inhibit bacterial adhesion to neurons. The next step is to load the drugs into the vesicles so that we can use the vesicles to target neurons. And then we could see that the vesicles with the drugs can very significantly inhibit bacterial adhesion to neurons compared to neurons without the treatment with EVs. And then importantly, empty EVs without the drugs, they don't lead to any reduction of bacterial adhesion to neurons. In parallel, we are also using EVs to directly block the beta-actin. So we are using EVs that have are coated with an anti-beta-actin antibody. And in adhesion assays, we could see that the vesicles coated with the antibodies could very significantly inhibit bacterial adhesion to neurons. And this impairment was actually more significant than when using anti-beta-actin antibodies alone, which means that the presence of the vesicles can really enhance the bioactivity of antibodies to block the binding of the bacteria to neurons. And uncoated vesicles, they don't lead to any reduction in bacterial adhesion. Then the next step is to add an additional coating on vesicles so we can target neurons. We are using an antibody against the ALK, which is a protein that is specifically expressed on neuronal plasma membrane. And then to show that this additional coating can really target neurons, we used these double-coated vesicles, and we treated human neurons that have ALK, and then endothelial cells HPMEC, which do not have ALK. And then these vesicles could bind well to neurons, and then we could see ALK protein band in Western blot, and then we didn't see the band in HPMEC because these cells, they don't have ALK. So this means that with these vesicles, we can really target specifically neurons. And last but not least, we could also show that with these treatments, the neuronal cell viability is not altered. In fact, we did cytotoxicity assays, and then we could see that with the triple drug cocktail to reduce beta-actin exposure, and also with the vesicles coated with anti-beta-actin antibody, the neuronal cell death is extremely low, almost compared to basically untreated neurons. So this means that both the approaches are not toxic for human neurons. So why we think that this bacterial neuron blockade strategy can be of a huge groundbreaking benefits? Because when a patient is hospitalized for a suspected diagnosis of bacterial meningoencephalitis, this is day 1, then is normally treated pharmacologically. If the symptoms are not going away, then there is normal, the second day, there is a lumbar puncture performed for CSF collection. And then there is a culture of the CSF in order to assess whether there is a positive diagnosis of bacterial infection, which normally comes between day 2 and day 3, because the culture of the CSF takes time. And then at day 3, there is antibiotic treatment starting, usually with beta-lactam, to treat the streptococcal infection. But as I mentioned earlier, because of the poor penetration across the blood-brain barrier, and especially the constant threat of antibiotic resistance, then most of the times this antibiotic treatment is not very successful, and therefore at day 4 there is a multiple antibiotic treatment starting in order to enhance the capacity, the possibility of the treatment to be effective. The problem is that during these 3 to 4 days, which seems a very short time, there is a constant neuronal damage. And neurons, besides, despite many other cells in our body, repaired neurons, they cannot be neither repaired or replaced. Therefore, the possibility to have a treatment that can block bacterial interaction with neurons can really solve all the damages that antibiotic resistance can cause during this time. And then this treatment in all these steps of these 4 days can really either prevent or minimize the onset of neuronal damage, which are the pathological hallmark for all the neurological sequelae that the people suffer after recovery from brain infection for the rest of their lives. So with this, I would like to thank all the members in my group and our collaborators at Karolinska Institute and the funding that make our research possible. And thank you very much for your attention, and I'm happy to take any question.
Thank you, Federico. A very, very exciting projects that you're running. If you have any questions to either of the speaker, you can raise your hand. You can write questions in the Q&A. In the meantime, I'll go back to the presentation to end our session. Gabriel will help you with questions as well. So please interrupt me, Gabriel, if needed. But today we run the first seminar of the fall. Next session is September 20th in the afternoon, that time. You will hear from 3 different projects, 3 different countries, all working on their specific challenges to fight antibiotic resistance. The link here will lead you to a registration page. So if you copy the link, write it down, you will be able to sign up. And also you find us in our social channels for the upcoming seminars. With that, I'd like to thank you for joining today. If there aren't any questions to Federico or the other speakers, I'll stay silent for a minute. In that case, if you have any questions, you're always welcome to reach out to me or Gabriel. We will be sharing material from the session in a capacity that the speakers allow. So some of the content you will have to have been here to see. Other content you will be able to review and take in afterwards. But with further ado, thank you so much for joining today. I hope to see you again sometime during the fall. Check out the calendar for sessions that you're interested in, and have a lovely rest of the day. And again, thank you, Federico, Ibrahim, and Zeenatuddeen for joining us. Big thank you for that.
Thank you, Cecilia. Take care. Bye-bye.
Have a great day. Take care.
Too.
Thank you, everyone. Bye-bye.
Bye.
Thank you. Bye.
Thank you.